Uh, Tonight's theme is COVID-19 research and drug development at SLU. Our speakers are Dr. John Tavis and Amelia Pinto. John is professor of molecular microbiology and immunology at St. Louis University, and Amelia is an assistant professor in the same department. John's research focuses on antiviral drug development for hepatitis B. He is studying how the hepatitis B virus replicates in order to develop a new drug that, in combination with other medications, could cure the viral infection. So today, John will be telling us about challenges of drug development during the COVID-19 pandemic. So please take it away, John.
Okay, and we can get started now. So my topic is challenges of drug discovery during the COVID-19 pandemic. This is a more of a general topic is what I'm going to be going through here. I'm speaking now as the director of the St. Louis University Institute for Drug and Biotherapeutic Innovation. This does not reflect my research. You'll hear more about from a personal perspective on research for COVID-19 from Amelia in a few moments. IDBI is a relatively newly formed distributive institute of drug discovery. There's 70 PIs and senior investigators at St. Louis University spread through all of St. Louis University's campuses and through a great number of our different departments that work collaboratively to try and accelerate drug discovery and biotherapeutic development at St. Louis University. If you have any questions about the Institute, I'm the primary contact on that. Please feel free to send me an email. I'm not hard to find by Google. Okay, so I want to start out talking about key issues affecting drug discovery, so just so we know what all the topics are that we're going to go through here. Then I'll go through each of these topics individually, how they relate to SARS-CoV-2, which is the pathogen that causes COVID-19, of course. And then the last topic, ethical aspects, is really a series of questions. And I'll go over some of the key issues of that, and hopefully that will set off a few questions from the audience to have a bit of a discussion going on. So that's my plan here. So the first one is, how does the biology of SARS-CoV-2 affect its spread and drug discovery? First off, of course, is its infectiousness and route. This is a very highly infectious virus, primarily by by respiratory droplets and then secondarily by fomites. So it can get all over fast, which is indicated by how rapidly it exploded across the world scene. It is a zoonotic infection. It came from animals. The trace of the animal developed evolution and transfer into humanity is not fully known yet, but it is clearly a zoonotic infection. Actually, many of the viruses that come out causing pandemics are actually zoonotic in origin, so this is not rare at all. However, very little is known about how the zoonotic aspect of SARS-CoV-2 biology is going to affect how the pandemic plays out. You know, for example, it can infect great cats, but I don't think the tigers are really going to be a major animal reservoir for them. And so, but as far as how this affects the drug discovery is it raises the possibility there may be a market for veterinary SARS-CoV-2 drugs. I personally think that's going to be quite unlikely, but it is possible and something to keep in the back of your mind if you're interested in drug discovery. The infection time course, of course, for this virus, it's moderately fast. It's slow, for example, relative to a norovirus, and it's lightning fast compared to what I'm used to working with hepatitis B virus. The key issue on here that affects drug discovery is that the infection does proceed slowly enough so that it is practical to diagnose the infection and start treating somebody before, early enough to have some therapeutic benefit. My guess is, and early indications in the early clinical trials are indicating that the intervention window is likely to be about 2 weeks from primary infection, given the median onset of disease symptoms is typically around 5 days, although it can be much slower. That gives you a good window for therapeutic innovation. And in the clinical trials with remdesivir, treatment concurrent with diagnosis in the hospital was enough to yield clear evidence of therapeutic benefit to the patient. Lethality of this virus, the case fatality rate is not known primarily because we don't really have an idea how many cases there are in the first place due to underdiagnosis and underdetection. But it's clear that it's relatively low compared to other pandemic viruses, such as the 1918 flu viruses, variola virus, which caused smallpox, Yersinia pestis, which caused the plague in the Middle Ages that killed a third of all human beings on the face of the earth. Yellow fever virus historically has had a 20 to 30% death rate once it got to its severe stages. So compared to some of the pathogens that nailed humanity in the past, this one isn't so bad. However, the lethality is moderately high in hospitalized patients. And so that has implications on drug design, because that means that you can tolerate some side effects in the drugs and safety concerns in the drugs, because you're dealing with something that is really got an urgent problem going on. Basically, we don't need to get a perfect drug the first time out. We can tolerate some problems with the drug. And for example, remdesivir has got some significant issues with it. Persistence in the human population, that's pretty much a black box right now. If this behaves like a typical respiratory infection, it is not going to be high at an individual level, but in the population level, it'll be quite high. Susceptibility of the host population to SARS-CoV-2. You have to break this into two parts. One is susceptibility to infection. That's very high, and that's due to a lack of herd immunity to an antigenetically distinct pathogen that's never been seen in the population before. This thing is ripping through a naive population. Susceptibility to disease is different. It is moderate. It's clear that the large majority of the cases do not lead to serious disease. But the ratio of asymptomatic to symptomatic cases we really don't know. But the susceptibility to disease and the susceptibility of infection combined to indicate this is going to have a huge market size, which of course influences how drug companies look at distributing their resources in development. There is a serious variability in disease incidence per infection. It's extremely poorly understood. It's affected by host genetics, immune state, diet, comorbidities, phase of the moon, Lord knows what else. Shoe size might fit in this. We really don't know. And so that, of course, it raises serious question marks in clinical trial design and the potential of designing personalized treatments for those people who are most at need of it. So that's a complexity you have to deal with. Durability of post-infection immunity. I don't think I'm the only person in the world who really, really, really wants to know what that is. It is unknown at this point. There is probably some post-infection immunity once you clear the infection. Obviously, your immune system's won in the first place, so it's capable of doing it. But we really don't know what it is, and this has a major impact on the shape of the pandemic, which in turn has major impacts on the durability and size of the market for a drug, which is something that the drug companies pay exquisitely close attention to. So disease manifestations affecting drug discovery. The diagnostic value of early symptoms can be really valuable. That was really, really important, for example, in clearance of the smallpox pandemic and driving the virus to extinction. Here, it's really low. I don't know how many times I described symptoms of a viral infection to the med students during the lectures as, quote, flu-like. And so it's really got a low early diagnostic value just from the symptoms. That clearly mandates development of very rapid molecular tests for accurate diagnosis and treatment monitoring, preferably point of care. There's an awful lot of work going on in that area. There's been some very, very good advances in that area, but we really aren't where we need to be yet. So there's plenty more room for those of you who love developing assay kits and tests, dive in and go for it. The major pathogenic symptoms, of course, the ones at least leading to death are primary viral pneumonia, and that mandates drug formulation delivery to the lungs. That doesn't mean that's the only place you want that drug to get, but it means it does have to get there. And it may, of course, permit development of symptom targeting drugs. You know, if you can keep the patient alive by just keeping the symptoms under control so that you can allow the patient's immune system to do the heavy lifting, well, that's good too. And then, of course, timing of infectivity onset relative to symptom onset is always a big issue in the spread of pathogens in a population. In SARS-CoV-2, it's clear that infectivity can precede symptoms in many cases, allows asymptomatic spread, and really complicates control by quarantine, which of course increases the need for vaccines and drugs. And this also, of course, may open a market for prophylactic drugs in high-risk population, like, for example, our colleagues in the infectious disease wards. But however, prophylactic drug use mandates that the drug be really, really safe, and that we know for sure it be really, really safe. And it's going to take a while for the clinical experience with the new drugs to come to the point where we can really be comfortable with prophylactic use, although there is clearly a market for it, at least until a vaccine comes along. Geographical distribution of SARS-CoV-2 is affecting the shape of the pandemic. The initial detection was found in Wuhan, which is a modern city with an adequate scientific medical infrastructure. I actually have been there. I've given a lecture at their largest infectious diseases hospital. It isn't quite up to the standards of the SLU or the WashU hospitals, but you're a long way from being out in the middle of nowhere. That clearly permitted early identification. Politically, some of that value was squandered, as we all know from following the news. However, it did give time for us to begin drug development efforts very early. Same thing goes for vaccine development efforts. And fortunately, the research community didn't mess up as badly as the political community did. So there's, right from when it became apparent that this virus was going to be a problem, a lot of companies and a lot of academics started diving in hard, trying to get this going. So we got a leg up on it. And of course, early penetration of the pathogen into developed areas such as Wuhan provided researchers with good and rapid access to patients and samples. That is, of course, always an asset. For example, the SARS-CoV-2 genome was published lickety-split in basically no time at all, which was obviously very, very useful for a number of things. However, the flip side of that is rapid penetration to areas of major research expertise, such as the New York City area, has hampered development by slowing down the ability of the researchers to do their job. Market location is really, really important. This one's worldwide, basically everywhere. So the first world markets can afford drugs. That's really important in the calculus of the drug companies because they need to be able to make their money back from their developmental efforts. Lower income countries are going to need cost adjustments and or governmental purchases in order to make the impact that we know we need to make. And a big issue is IP protection in lower income countries is at serious risk due to the extreme urgent need that those countries have, which of course has major financial implications on how this is going to play out and what the pricing spectrum is likely to look like. Okay, state of preparedness for SARS-CoV-2 is also a really important topic. Relationship to known pathogens, that's a big one. SARS-CoV-2, I should have a 2 there, sorry, messed up there, typo. Is very similar to the seasonal coronaviruses, MERS coronavirus, and SARS coronavirus at the big picture. I mean, it's a coronavirus. We know stuff about coronaviruses already. We'd love to know an awful lot more to get to the nauseating level of detail that's needed for drug discovery, but we're not working from a completely blank book. Okay, we know what a coronavirus looks like and how it works. Repurposing existing drugs or drug candidates is absolutely essential for early success. Drug and vaccine development, clinical trials, related pathogens really laid the groundwork for rapid drug development. Remdesivir, for example, is in clinical trials for Ebola virus. Favipiravir, and I'm always really bad at pronouncing that, is an approved drug in Japan for influenza virus. And so these are the two leading drugs. Remdesivir has shown some clinical benefit. It's not where we want it to be yet. It's got some clinical benefit, and that was able to be done that quickly because the safety studies were already done. We knew how to dose it. We knew what the human doses needed to be, so we could move almost instantaneously into a clinical trial. One of which was run at, one arm of which was run at St. Louis University. And I noticed when logging on that the local PI of that, Dr. Sarah George, is one of the attendees at this webinar. Without such hypothesis-driven repurposing, new drug discovery's got to start from scratch, which means we're not going to get anything quickly. You know, the average time of development from concept to first sale is about 10 years. And obviously we don't have 10 years right now. We got to move faster than that. So that means repurposing is the name of the game. Funding for drug development for emerging pathogens always stinks until an outbreak hits. And then money gets poured on it like crazy, and then it dries up immediately afterwards. SARS-CoV-1, which caused, I believe, what was it, the 2002 epidemic, is a good example of that. You know, a whole bunch of money got dumped into it, and then it dried up. That's likely to be what's going to happen here also. Fortunately, however, SARS-CoV-2 drug development and vaccine development, by the way, falls in the same category, has benefited absolutely, tremendously, enormously, bigly, hugely, however, whichever adjective you want to use, from the investments in the SARS-CoV and MERS-CoV emerging viruses. You know, we have clinical trials going on right now of vaccines against SARS-CoV-2, and those, we couldn't have gotten anywhere near that area if we hadn't had vaccine candidates for SARS-CoV and MERS-CoV. And so rapid allocation of government and corporate funds for drug discovery is enabling rapid progress, and that really is, there is an enormous amount of very rapid progress going on right now. So finally, the last slide I have here, and this is more to get people thinking more than to say that I know all the ethical answers, because I don't. But under drug trials, I mean, how do we accelerate the trials safely? I mean, adaptive trial designs, we've got to really rock things along here. But how do you design a control arm when you're dealing with a deadly disease for which there are no treatments? How can you manage compassionate use? You want to do it on one hand, but it messes up your data for determining whether or not the drug is working properly. And it can confuse the messaging into the population, and that has been clearly a problem for us here with SARS-CoV-2. When is a challenge trial ethically permissible? You know, challenge trials really move it along pretty quickly, but ethically giving somebody an infection with a lethal virus when there's no treatment for it, even in low-risk populations, has got huge problems with it. There's been something like 14,000 people who volunteered for a challenge vaccine trial already, but getting that past an IRB is not going to be easy. That will get much easier for vaccines if it turns out that some of these repurposed drugs that are being tried actually have some decent efficacy, better than remdesivir. Regulatory control, this is a huge issue. You know, how flexible and responsive is the regulatory agent? How can intellectual property be protected? You got to be able to get the enormous amount of money being dumped into drug and vaccine development back. And how overlapping and fragmented is the regulatory landscape? And the answer is it's very fragmented on the world stage. Distribution, big questions. Why don't you watch the movie Contagion? You get a really good sense of the complexities of deciding who gets the drug first, who decides who gets it. What's your goal? Are you trying to control it locally, regionally, nationally? I mean, the rise of nationalism is getting people saying, I want this, this is all for mine, but we really need an international treatment capacity. Who goes about delivering it? These are just nasty questions. Pricing, who pays? How does the company recover its costs without appearing to be a vampire? There's already been blowback on Gilead over remdesivir, over its pricing. You know, how is the pricing determined? How variable is it, and what special populations need special financial considerations? So these are all really big, knotty questions. So with those questions, for which I do not have answers, I will end. and take any questions if anybody would like to chime in.
Well, thank you very much, John. And very interesting talk. And I'm sorry to hear that you don't have answers to those ethical questions. But we'll open it now to questions from the audience. So people are a little shy often. And so just to get started, I wondered some of these issues that you raised earlier about repurposing other drugs. Do you expect that to be highly successful, or how rapidly do you expect that to be modified something that that already
Well,
exists?
I don't play the lottery, and being able to predict the outcome of the lottery ball drops is about as good as you can do here. Until we get to clinical trials, it's hard to say. What I can say is basic biology says that we know what types of drugs you'd like to deal with. I mean, an entry inhibitor, for example, that binds to the receptor, both from remdesivir and favipiravir. And again, I apologize for the massive mispronunciation. I see Amelia's laughing at me. Those are both RNA-directed RNA polymerase inhibitors. There's a good track record for RDRP inhibitors in clinical trials and in approvals. For example, sofosbuvir against hepatitis C virus is an extraordinarily effective drug, and it works by the same mechanism. Basically any of the essential viral enzymes are obvious targets, and so we're not running completely blind. We know which sandboxes to play in, but those are big sandboxes.
Oh, and we have somebody who's raised a hand. So Andrew.
Yeah, I was wondering about the drugs. What about like using the flu antiviral drugs and the HIV antiviral drugs? Are those of any possible use?
Well, the HIV ones are probably not. HIV is a reverse transcribing virus, and its pathology is very different. The cell types it gets into are largely very different. So I was really surprised when they started talking about them. The limited trial data that I've seen, the HIV drugs that have been tested flopped miserably. For flu, you got a different topic going on here. Flu causes similar pathology, infects similar tissues, has a similar overall cellular and viral replication cycle. And so their drugs like favipiravir, they actually have a decent chance. And favipiravir, and again, I apologize for killing the name, it works really, really well in cell culture. Okay, so it clearly works against the virus. The question is whether or not it's going to get where it needs to be in the body fast enough and at a high enough concentration to do what it needs to get done. That we don't know, and only clinical trials can say. Oh, the other flu drugs, no, they work by flu-specific mechanisms. So no, you're not going to have Tamiflu working against SARS-CoV-2.